Hi everyone, this is Rachel King, and in this recording I'm going to talk to you about experimental design. So in the previous recording on data analysis, I briefly touched on two main types of research that can be defined by the way the data is collected: experimental and observational research. So to go into a bit more detail, experimental research generally has a treatment that's applied to the sampling units within your research. So if you're doing a study on how fertilizer affects plants, your units of study are your individual plants, and some of them may receive a fertilizer and some of them may not. So the treatment is the fertilizer, that's the manipulation you're putting into place, and you generally also have a control group where you don't apply that treatment. Often you can have more than one treatment, so you could be trying out several different types of fertilizers. So the advantages of an experimental design are that it's repeatable. The methods are clearly set out and can be reapplied to another set of sampling units if you want to. And because of the way it's designed, it is possible to try and control for unwanted variation, so that you're actually measuring the variation between the treatments that you're applying, and this variation is not being confused with other differences just between the individuals within each of your groups. It is only with an experimental design where you have a control group and you have applied, the researcher is responsible for actually applying the treatment. It's only in this kind of research design that you can then, from the data, establish any cause and effect. And that is precisely because in this type of design we have tried to control for as much of the unwanted variation as possible. And so we know that any differences truly are caused by the treatment that we have imposed within the experiment. The disadvantages of this kind of research is that it's often quite expensive and time consuming, and if it's involving people or animals it can require ethics. One of the primary issues can be that because it is time consuming and expensive, the number of replicates, which we will talk a bit more about in a moment, can often be limited just by the cost, which is not always the best approach. So an observational study, in contrast, can also seem experimental. There can also be some kind of manipulation or treatment applied. But in an observational study, it's not the experimenter or the researcher who's applying the treatment. So if we think about people who smoke, and if we wanted to look at their lung capacity, we could have a group of people who are smokers and a group of people who are non-smokers to compare lung capacity. But those smokers would have to be what we call a naturally occurring group. They chose to smoke. They're already smokers. They put themselves into the smoking group. It's not the researcher who has made them start smoking just for their experiment, because that would be unethical. Surveys are another form of observational study, but in a survey, rather than directly measuring some characteristic of each of the individuals in our study, we are actually measuring their opinion or their feelings about things or their perceptions of things. So it's not necessarily as direct a measurement process. Often in a survey, the kind of scale we would use to measure people's opinion is a strongly disagree to a strongly agree kind of scale. And within that, there's often quite a bit of variation in how people perceive the changes from one category in that scale to the other. So my perception of the difference between strongly disagree and neutral might be different to yours. Some of the advantages of a survey: it's possibly quicker and cheaper, especially nowadays when you can have online surveys. Some of the disadvantages are it's less repeatable, you can't control for extraneous variation, and you definitely can't establish cause and effect just based on a survey. One of the other things I would note with surveys is that researchers are finding it harder and harder to get people to actually fill out their surveys. People are over-surveyed in the general population, and they don't really appreciate being asked to fill out a survey. So you need to put a lot of thought into the design of your survey if you're using one for your research. You don't want it to be too long. You want it to get to the point. The person who is completing your survey needs to feel like it really does relate to them and their life or their work. Otherwise, what you end up with is people who start a survey and don't finish it or leave missing data, and that makes your analysis later down the line much harder. So why should you worry about design and analysis? Is it important for your research? Well, I'm a statistician, so obviously I think it is, but I think also that it's important for you to understand that your interest in research is probably driven by your interest in your discipline area, whether that's health, environmental, or computing, or business research. It's your interest in the discipline that is driving your interest in research. However, as a researcher, you can collect as much data as you like, but unless you really think about the design of how you're collecting that data and then how you're going to analyse it, all you are doing is collecting information that's descriptive. It doesn't help with decision making within your discipline or field. It just becomes a descriptive process where you've gone and collected all this data and then you're going to try and summarise it and describe it, but you can't really analyse it in a way that's going to improve your understanding of the features or characteristics that you've measured in a really much broader context, so beyond your sample and into the population. So as a researcher, design and analysis are critical aspects of the research process. So typical steps in experimental design or really research design is that you have some background knowledge, and this is where researchers start. They have some interest or curiosity about something within their discipline after reading some background research or previous work. And then from that they sort of come up with some really general questions about that can be sort of really convoluted and interwoven kinds of questions. The important thing is to break those sort of general questions down into very specific questions that can be answered. General long-winded paragraph-style questions really are hard to address in a practical sense, so you need to break your general questions down into really specific questions. From those specific questions, we form hypotheses. So the null and the alternate hypotheses, they're statistical terms. They relate directly to the type of analysis you're going to perform. So you have different types of hypotheses for different types of statistical methods. Each type of method has a null and an alternate hypothesis that should be stated, but the form of those hypotheses can change depending on the method you're going to apply. And the method you're going to apply will depend on the types of variables you have measured. So there's a real interconnected link between these parts of your research approach. From your questions, you need to be able to come down to really specific questions, and from those you need to be able to directly link to measurable variables. So any specific research question you have, you need to be able to identify how you're measuring variables that will let you answer that particular question. And the form of those variables, whether they're categorical or whether they're continuous or whether they're nominal or ordinal, the form of those questions will then help you or will guide you in some ways into what kind of analysis you'll be able to do and how you should form your hypotheses. So we get to the design of the experiment. Once you've got your questions, you need to design your experiment and figure out how you're going to measure things. You need to run the experiment and collect your data and do your analysis and then come to your conclusions, and then that becomes background for the next round of research. So as you would have noticed as I was talking about these earlier steps here, this really is not a linear process. It's not straight through. You will have your questions, you'll get specific questions, you'll frame some hypotheses, you'll get to the design stage and realise you can't actually measure some things you thought you would be able to. Maybe it's too expensive, maybe you can't get ethics approval, or something like that. So you need to rethink the questions you can ask. There's no benefit to just push ahead and collect data that isn't going to be able to answer your original questions. So if you find that at the design stage that there's conflict between the design you can run, that is practical to run, and the questions you had, you may need to reframe these questions. Then you run the experiment and collect the data, and hopefully everything goes perfectly well, but most of the time something crops up that was unexpected. And this again may need you to either drop one of your questions or reframe one of your questions. And so again, we're looping back to the start and then coming through again. This process through to analysis can go perfectly. Nothing goes wrong. Everything is exactly right. You have great, responses to your survey, or nothing goes wrong in your experiment, and you've got really great, perfect data. There's no missing values. Everything's really good. That still doesn't mean that your analysis will lead to the conclusions to these questions that you thought you were going to get at the start. There's no way to guarantee that the data you collect will give you the results that you hope for. There's no planning for that, because what you measure is what you measure, and maybe what you hypothesized at the start is just incorrect. Maybe what happened is that the population you sampled wasn't actually the population you were thinking you were sampling up here. So when you get the data and you run the analysis, you realize actually your sample represents only a small subset of the population you thought you were collecting data on. So again, you may have to reframe. So this slide is also trying to point out the sort of non-linear, really. It's not really a linear process. You're going to be looping back all over the place. That may seem a little daunting, but I would suggest actually that you just embrace this process. It's much better to take the time to plan and then replan and then really be on top of this process, even if it does seem a little bit exhausting. It's a much better approach than just pushing through, barreling through in a linear way and then getting to the end and trying to force conclusions out of your data that your data doesn't really hold up to. I'm just pointing out here, these are statistical terms when we're talking about the null and alternate hypothesis. When we have our research question, our research questions may be, I think there is a difference in the average heights between males and female students studying Sci 4405. That's my research question. I think it's worth me collecting this data because I think there is a difference, and I think that difference in heights might be really interesting for some reason. So when I'm framing my null hypothesis, we frame the null hypothesis in statistical terms to state that there is no difference. The null hypothesis would be that there is no difference between the average heights of my male and female students, and the alternate hypothesis is that there is a difference. So your research question is really the alternate hypothesis. Now we frame them that way, we frame our statistical hypotheses that way because when I collect the data, based on probability, I can conclude that I have enough evidence in my data to reject the null hypothesis, that they are not equal, and therefore I will accept my alternate hypothesis. So I reject that they are equal, that there's no difference, and accept that they are different. I can never prove that they are different. Just to a certain level of significance, I can claim that I have enough evidence to suggest that they really are different. So some things to consider about experimental design. So your methods need to be explicit, consistent, and repeatable. Repeatable is incredibly important. Science does not move forward based on one study. So the only way our knowledge improves, our knowledge about the world improves and becomes more and more reliable is when a study is repeated and finds very similar results. So going back to my example of measuring the average heights of male and female students, I couldn't make any general claims about that based on one class in 1 year. I would need to repeat that, and then after 5 or 6 years, if I kept finding a similar amount of difference between male and female heights, I could then start claiming some real substantial evidence that there's a general trend of this difference, but not based on just one study. So as I mentioned in the previous recording, statistics is about understanding variation, and there's different forms of variation. So there's inter-entity variation. So in my example of heights of students, the inter-entity variation is the difference between individual students. So we know that there is going to be variation among individuals. What I'm trying to research in this question is whether I can claim that that difference is due to sex, whether the students are male or female. Now within the male and female group of students, I know that individuals will also vary. What I want to understand is whether I can make a claim about the group differences based on individual measurements. So inter-entity variation is the difference among the different students. Intra-entity variation is the differences or variation that may occur within an individual. So this is generally something that may occur over time. So height definitely in an individual person would change over time. So maybe it's the moment that I measured the person that is affecting my outcome. Pre-analytical variation is variation in experimental conditions. So height probably isn't a good example here, but maybe if I was measuring something that was affected by temperature or humidity or many other kind of environmental variables generally, and this is particularly applicable in a lab situation, if those pre-analytical variables vary over the time that I'm making my measurements, some individuals may be more affected than others. There's also analytical variation, and that generally refers to equipment differences. So variation due to instrumental differences. Perhaps if I'm measuring UV and I'm using multiple instruments, I need to make sure they're calibrated correctly so that I'm not introducing sources of error that will confuse the differences in the things I'm actually measuring and the effect I'm trying to measure. And then there's experimental variation, and this is the variation that I'm actually interested in. This is the stuff I really want to know. I want to know if there's a difference between the average height of my male and female students, so the group differences. So I want to try and limit the other differences that are going to maybe confound this. So the object of any experimental design is to highlight experimental variation relative to other sources. So you can think in an experimental situation where you're actually controlling environment, you're limiting all other difference except for the treatment you're applying. That is why in an experimental situation we can really claim cause and effect, because we've limited the other sources of variation and we're just really trying to measure this variation. You can imagine that if you were using a survey for your research rather than a clear experimental design, it would be very hard to control other sources of variation. So part of this process is to really think about how do you measure the variable of interest to best capture the variance that you're looking for. So that's using, measuring height. Well, the best way to measure that is with a tape measure. There's no better instrument available than to measure it with a tape measure. But if you're measuring other things, you need to really think about what scale, what measurement system you're using. And this, again, still applies to surveys. You need to really think about how you're forming your questions and what scale you're using to measure the variation. So one way to control for unwanted variation is to think about in an experimental situation how you're measuring your different individuals and groups. So say in this example we have two different labs and we have to use some different chemical reagents to measure whatever it is we're interested here. If we ran all of our control samples in lab 1 and all of our treatment samples in lab 2, how do we know that any differences between treatment and control samples aren't due to some difference in the different lab that we performed the analysis? So this is where we need to think about randomization. So in this situation, it would be much better to randomly allocate control and treatment samples to each lab, so that if there is an effect of lab, and then maybe the chemicals we're using in each lab, which may vary a little bit, or the temperature in the lab or something like that. If there is that source of variation, it's not being confined just to the groups that we are actually interested in the experimental differences or the experimental variation. This would also work even if we were just working in one lab and we did all of the control samples on day 1 and we did all of the treatment samples on day 3. That could also introduce unwanted variation in a way that would exacerbate the difference between our control and treatment groups, but not really be a true measure of the true variation between those groups. So variation can be thought of as either systematic or random variation. So by using two different labs, we are introducing a systematic form of variation. Random variation is the normal variation we expect between individuals. So we want to really avoid introducing any systematic variation that isn't actually our treatment effect that we're trying to measure the variation on. So how do we deal with these sources of variation? You may want to avoid them. So perhaps you're running an experiment on some mice and you think there may be variation between male and female mice that really isn't of interest to you, and it's just going to obscure the true variation you're looking for because of the application of your treatment. So what you may want to do then is redefine your population to just be about female mice, for example. So you're excluding some variation you think might just confuse the issue, and just narrowing down your population. That means that any conclusions you come to also are narrowed down to referring to that smaller population of just female mice. You can randomise effects, so that's as I was explaining on the previous slide, so not doing all of your samples in the lab on one day or in one lab and then the control in another. Or you can incorporate it into the experiment. So perhaps with the male and female mice example, you don't want to exclude the male mice. Perhaps you actually just want to include sex as a variable in your analysis. So that can mean a little bit more time in recording data. So now you have to record whether the mouse was male or female. You need to make sure that you don't have all the female mice in the control group and all of the male mice in the treatment group. You need to make sure you randomise the allocation to the groups across the sexes, but it is a way of retaining the full initial population and just incorporating that source of variation into your analysis. So your control should be a subset of your total sample. It's important not to select your treatment sample separate from your control sample. You should select your sample and then subset that overall sample into a treatment and control group. And remember, you must have a control if you want to try and claim causal effects. So here we have some fish, and we're going to add a poison. If we have no controls, we just have one tank of fish and we throw in some poison, how do we know that mortality is due to the poison or the toxicant and not something else? How do we know that these fish weren't really elderly and dying anyway? How do we know it wasn't something in the water before we put the toxicant in? We wouldn't know. However, if we take our overall sample and we split this sample into a treatment and control group so that they have exactly the same water, they have a similar combination of fish in each one, and the only difference is that this group gets the poison and this group doesn't, then we really can say that the initial variation in each of these replicated each other. So the only difference is due to treatment, and so any difference in survival we can then claim is due to the adding of the poison. So randomization is the process of randomly allocating individuals to either treatment or control group, and every individual has the same chance of ending up in either the treatment or control group. So you need a way to make sure it really is random. So say you had a researcher investing changes in acetylcholine A in lab mice, and they have a big tub of mice, and they select out the mice that are easiest to catch for the experimental group, and the others are in the control group. Or maybe they have this sort of subtle preference for black mice, and so those black mice end up in the control group, and the bitey, nasty ones end up in the experimental group. These researchers then introduced bias into this study. Maybe bitey, hard to catch mice are stronger, more resistant to whatever you're going to be doing to them, and you've sort of collated all of those kind of mice into one group. They're not evenly spread across your treatment and control groups. So you need to find ways, if you're running an experiment, to make sure that randomization or random allocation to treatment and control groups is really random. You can use random number tables. There's lots of different ways to do that, but the thing I want to draw your attention to is not introducing your own bias into that allocation. We all have unconscious biases. It's no individual flaw in anyone that they would allow their biases into their research. It's possible for all of us to do that unintentionally. We just need to be aware of that and try and put in place restrictions to stop that happening. So now I want to talk about replication. As we've been talking about statistics and analysis and research, it's all about understanding variation. So to understand variation, we need to measure things on more than one individual. We need to have a measure of variation, and that requires replication. We need more than one measure. So there's two main criteria for good replication: that the replicates must be chosen at random and that they must be independent. If you're doing blood work on two different mice, the levels of hormones in one mouse is not going to affect the level of hormones in another. Now, pseudo-replication can be a really big issue in research. It's where you've measured several variables of interest, and so you have repeated measures, but they've been made on the same sampling unit, so they're not independent of each other. And I'm going to show you a couple of examples of that. So in this example, we have a header tank that has some solution in it. And there's multiple header tanks, but what we've done is take one header tank and we've taken 3 samples out of it. So these 3 samples are replicates of just this one header tank. So if our research question is about the toxicant levels among header tanks, we don't have replication of that. We just have replication of this one header tank. What we should be doing is taking a sample from each header tank, or what would be even better and is often done in chemistry, is to take 3 samples from each one and then take the average of those 3 for this one as a measure. That's a typical thing done in chemistry because you get slight variations due to equipment or lab procedures. So you take 3 and you take the average. Another example is looking at an ecological context. So if we're looking at effects of fire on vegetation. So each of these green squares is a plot, and then within each plot we've taken a sample. So maybe we've thrown a quadrant on the ground and we've looked at vegetation cover or lack of vegetation cover within that quadrant. So if we go to one unburnt area, one plot, and we take 3 measures within that one plot of an unburnt area and again of a burnt area, these are pseudo-replicates. They're just 3 measures of this one area. They don't represent unburnt areas in general. If we went to 3 different burnt areas, so 1, 2, and 3, and we took a sample within each of those independent areas, and same for unburnt, then we would have true replication. On this one, I'm measuring some hormone levels in mice. And so I take a mouse and I take blood from it for 3 days. So this one, I think it's really clear that those 3 measurements are not independent measurements of hormone level. They're 3 measures of the mouse. If I want independent measures of hormone, I need different mice. Now, if your question is about the change over time in hormone levels, then this is correct, but we still need to have that 3 days on the 3 different mice, okay? Because even though we're interested in changes over time, I need replication of changes over time. So I need this mouse for 3 days, this mouse for 3 days, and this mouse for 3 days. Sometimes when you're thinking about your research and what you should be measuring and whether you're replicating properly, it can help to set up your Excel spreadsheet before you even have any data. Just think up what your Excel spreadsheet might look like, because that's how you're probably going to record your data. So if we look at this one, this was where we had pseudo-replication, because we had what we had was plot 1, which represented unburnt, and we had 3 replicates in plot 1, and in plot 2, plot 2 represented burnt areas, and we had 3 measures there. I've just made up these cover measures here. So really what you can see now is that in terms of burnt, I've got 3 measures, but they all came from the same main plot. So really these are just replicates of plot 2. They're not replicates of burnt areas. In this version over here, I had 6 different plots. These 3 represented unburnt areas and these burnt areas. And I had 1 measure here, 1 measure here, and 1 measure here. So now these 3 all represent unburnt, and they're all independent because they come from 3 different plots. This slide is pretty much just saying what I just said in the previous slide. So we've got these 3 are pseudo-replication, and these ones are independent measures. If we look at our mice, so you can see here I've got 1 mouse and I'm measuring on day 1, day 2, day 3. So, I don't have replication of mouse. Mouse is my sampling unit. So what I need is more than 1 mouse. Now I've got replication of day 1, day 2, and day 3. So mouse is the sampling unit, so we need independent samples of blood from different mice. This is the header tank example. So in this one here, I have set it up to show that we've got 1 header tank and then the second header tank. And then I've taken 3 sub testing tanks from the header tank, another 3, and then another 3, and I've taken 3 reps here. So what I could do is average whatever chemical I'm measuring within the header tanks. If I average these values here, then I would have one measure for header tank 1, second measure for header tank 2, so I'm going to average these, and they will represent an average for testing tank 1, and that represents one replicate for header tank. Then I'll take these, take the average, and that represents a measure for testing tank 2, and that's the second replicate for the header tank. So I hope you can see from this that by setting up your Excel spreadsheet, it can help you really plan out your research. Don't wait to do it until you've got data. Just start thinking up of scenarios of what your data might look like. Oops, sorry, okay. I'm going to talk a bit more about the observational types of research now. So I mentioned earlier on that you can have observational studies that look like experimental studies, but the treatment has not been imposed by the researcher. These are often called quasi-experiments or natural experiments. You might see these terms in the literature. So the membership of the treatment level is determined by the conditions beyond the control of the experimenters. Subjects are already in that box. The smokers are already smokers, so they're already in the treatment group. An experiment may seem like a true experiment, but if the subjects have not been randomly assigned to the treatment condition, then it's a quasi-experiment, and you can't claim cause and effect from this type of experiment. Surveys are another subset of observational studies. They're often based on convenient sampling, so they're the simplest, least reliable and cheapest form of sampling. You don't have a control group to ensure precision. Used really frequently in market research and public opinion polls. The survey instrument, so the actual survey with the questions, we call a survey instrument, can be used in repeated studies, but the non-random sampling can make it difficult to control for unintended effects and variation, so they limit the repeatability of the study. Disciplines like psychology have spent many decades, and a lot of their research has been based on making sure they have developed really good surveys, so they've been tested and tested and tested, and the types of questions that are asked have been repeated and repeated so that they know they're drawing out the variation and the differences amongst people that they're trying to. So I would think that psychology is probably the discipline that has done the most work on ensuring that surveys are really good measurement instruments. But any survey that you create yourself in your research that has never been used anywhere else, it's a good process to go through, and I don't want to discourage you from doing that, but you've got to understand that if it hasn't been tested over many different research projects, then you have to be cautious about your interpretation of your results. So in surveys, we often have groups of questions that are relevant to an overall theme. And so once we get the data, we may not want to analyse each question, the responses to each question individually. We may want to analyse them as groups. And so we can use different methods to combine responses to multiple questions on the survey. Sometimes what we can do is just add up and take, the average of people's responses to several questions and then compare that one combined question. In other cases, we need to use actual statistical methods such as factor analysis or principal components analysis to perform data reduction, which will combine groups of questions across the survey. How to best capture variance is a really big question and can be a really big issue in survey design. There is a tendency when people are creating survey questions, they know their field very well, and so they ask a question because they know that this particular issue is a big issue in their field. But when you think about the types of responses you get, you need to think about whether you are framing your questions so that everybody's just going to answer strongly agree. If that's the case, then you're not really measuring any variation in your sample or your population. All that's going to tell you is that everybody strongly agreed, but there's no differences within or among groups then. So on the study desk in the data analysis and experimental design block, there's a paper I've provided by Broman and Wu. it's 2017 paper in the American Statistician, and it gives a really good overview of things to think about when you're designing your Excel data spreadsheet. I don't think you need to follow all their suggestions all the time, but I think it's a good thing to read before you start out because it'll prevent lots of issues later on. You'll need to read that paper for the assessment item for this topic, so I think generally it has good advice. So some of the key suggestions they have is that if you have categorical variables with text values, code them. So if you've measured sex and you've got male and females, don't type male and female into the cell in your Excel spreadsheet. Start coding things, males are 1, females are 2, or vice versa. And make sure you have a good sheet where you're listing all your codes so you don't forget and swap halfway through. This will make any analysis, whether you're using SPSS or R or even in Excel, it will make your analysis easier. Use consistent codes for your categorical variables, and if you have missing values, make sure you put either just like a dot or something in that cell so that as you're entering your data, if you have to enter it from hard copy into an Excel sheet, if you just leave a cell empty, you will come back in a week's time and go, oh, did I miss something, or is there something meant to be there? So you'll do a lot of rechecking. If you put just something in that cell, like a dot or an X or something, then you'll know that it's a true missing value. Don't use zeros for missing values. That will cause a lot of confusion later on in your analysis. Don't have symbols or spaces in your variable names. There's no analysis software that will correctly input this data. Most of them will have issues at least in some occasions. So if you have a variable name that is height of person, then either just use height or link the words by using underscores. Otherwise, it will treat each word as expect a new column of data for each word in your heading. Use a consistent date format, and also think about data precision. So by that I mean think about how many decimal places or significant figures you need to include. So if you're including 4 decimal places in any values you're putting in, that's indicating a really high level of precision, that you're sure of that value up to 4 decimal places. And that may be sort of overstating your measurement instruments a little bit. It may be correct, maybe that's what you need to include, but really think about that and be consistent throughout. Have only one thing in each cell, and make sure your data sheet is rectangular. So you have your cases, so your individual mice, or whatever you are actually measuring, have those in your rows, and the variables, the thing you're measuring on each of those individual cases, have them in your columns. Don't have any calculations in your Excel spreadsheet if you want to import that sheet into some other software like SPSS or R. Make sure it's just the raw values. And don't have any extraneous things like color-coded cells or anything like that on your import sheet. And also please make sure you back up. There's nothing sadder than seeing a research student who has lost all of their data and all of their analysis. Make sure you have a good backup plan for your anything you're doing on your computer. So whether that means you have to go and buy a backup drive or you're backing up to the cloud, just make sure you have something in place. Talking specifically about data analysis now, data analysis can be descriptive or inferential. So descriptive is describing your sample, maybe using counts or percentages or means or standard deviations or some other simple statistics. Inferential analysis means that you want to make conclusions about your sample and infer from those conclusions conclusions about the population. So inferential statistics means that we have applied some inferential method where we have a specific hypothesis that we've either accepted or rejected the null hypothesis. Remember when we say we're accepting a null hypothesis, that's not saying we've proven the null hypothesis true, it just means we don't have enough evidence to reject it. So data analysis can range from simple graphing of data to high-powered or complex statistical and numerical methods. Generally, when we're thinking about data analysis, the first thing we do is, no matter what our ultimate plan is for the data, is we do some descriptive analysis. We do some graphing or plotting and summary statistics. 1, to check our data, to clean it, to make sure there's no problems, and 2, just to get a better understanding of our data. We may need to do some data reduction. So if we've got a lot of missing data, we may need to remove variables from potential analysis because there's just not enough data there. We may need to remove individual cases, so individual sampling units or people or mice or whatever we've been measuring, we may have to remove individuals from our data if we don't have enough data on individual cases. Hypothesis generating and testing. Generally speaking, hypothesis generation occurs if you have collected data with no clear research question and you are using that data to help you generate a hypothesis. That kind of approach to research should be seen as a pilot study. So you're collecting a small amount of data for hypothesis generation. Usually what should happen in a research process is that you have a clear question. That clear question directs you on what kind of data you need to collect, and you're going to test clear hypotheses. So it's for hypothesis testing processes. Once you have tested your study's hypothesis, you can use that data for future generation of future work, future hypotheses. But if you are going out and collecting data with no real plan and then just using it as data mining, there's a real problem with that in terms of applying statistical methods. At some point, you're going to find significant results just because you keep mining the data more and more and more. I'm going to talk about that a bit more in the next few slides. It's called p-hacking. It's becoming more and more common as we have really large big data sets available to us. But you have to be really careful with it, how you apply that kind of process of just using data for hypothesis generation and then using the same data to test the hypothesis. We need to be really careful that we don't start falling into the trap of using data for hypothesis generating, just mining it until we can see some kind of pattern, and then using that same data to test the hypothesis. That's not a good way to perform scientific analysis. So as I said, there's really two forms of data analysis: exploratory, which is descriptive, maybe reduction, data reduction in there as well, and some hypothesis generation, and confirmatory or inferential, which is hypothesis testing. So inferential data analysis is traditional statistics designed to accept or reject a null, explicitly stated null hypothesis. So in that process of testing the hypothesis, we state the null hypothesis, we apply the appropriate test statistic and compute that test statistic, and then we find the p-value associated with that test statistic to determine whether we can reject or accept our null hypothesis. But I want to caution you against finding the p-value as the ultimate goal in your statistical analysis. Statistical significance, or the p-value, just means the result is unlikely. So if you have a p-value less than 0.05, 0.05 is used as a general standard of statistical significance. Finding a p-value of less than 0.05 just means that the result you found is unlikely if the variation in your data is just due to random chance. Therefore, we can conclude that it's due to the effect or the groupings that you have been testing. So the p-value is the probability of seeing a particular effect or larger if there is no real effect at all. Remember, our null hypothesis states there's no difference between groups, or there's no relationship among variables. So if we have a small p-value, it's saying if that was true, the chance of seeing the pattern in your data that you're actually seeing is so small that we're going to say that that's false. There is really a pattern, or there is really a difference. But the p-value itself, finding a significant result, doesn't tell you anything about the effect size. If you've got small variation within two groups, then you're more likely to find a statistically significant difference between the means of those groups. But if you've got lots of variation within your groups, it's going to be harder to find a statistically significant difference between the means of those groups. So we always need to make sure we report not just the p-value, not just that something is significant, but we need to report the actual effect size that we have seen in our data. We should also remember that the general rule of a p-value being less than 0.05 as indicating significance is just a rule of thumb. We can't treat it like a black and white rigid line. Significance levels were developed by statisticians at a time when computers were not available, so they came up with general standards and built significance tables, statistical tables around those general standards of 0.01, 0.05, 0.001, those kinds of things. But now when you run an analysis on your computer, you won't get information out that says your p-value result is less than 0.05. You'll get the exact p-value. You'll get a number that is 0.003, for example. So you can report the exact p-value and not just say that it's less than 0.05. But if we try and treat 0.05 as some kind of black and white decision-making rule, we can come across real problems. So this is an example of how just increasing your sample size can affect p-value, and why it's important to always report not just the p-value but the effect size. So here we have two groups. One has a mean of 10 and one has a mean of 12, and they both have the same standard deviation, which is a measure of the variation. So if we've got a very small sample size, this difference of 2 in the means would not be significant. So here's 0.1, 0.01, so 0.05 sort of around here. So unless our sample size is getting up past probably around 100, we would not be getting a significant result. But if we have sort of around 500, we have a very highly significant result. But nothing has changed. The difference between the means is exactly the same, and the variation within each group is exactly the same. Now, mathematically, this is exactly what we want to happen. If we've got a larger sample size, mathematically, we want to be more confident that this difference of 2 is actually because of the group membership and not just because of the sample we've got. Our sample sizes are big enough that we're confident that this difference is due to the difference in group membership and not just variation within each group. So although mathematically this is correct and this is not a problem, it's not a mistake, it's not something to be worried about, in practical terms for a researcher, what it means is you have to be really careful when you start interpreting p-values. So you have to be able to describe that although this is a highly significant result, you as the expert in your discipline needs to interpret whether a difference of 2 is actually practically meaningful in whatever variable you've measured here. So if this is a difference of 2 centimetres in tail length of a mouse, is that in any way meaningful really for the question you've been asking, even if it is significant statistically? So a large p-value is not proof of no effect, and a small p-value is not proof of large effect. So most forms of statistical analysis are classed under a grouping of parametric analyses. So this is where we estimate from the sample the sample statistics, and from those we infer the population parameters, so things like the mean, the variance, the regression coefficients, etc. To do this, analysis makes a number of assumptions, typically that the population is normally distributed, samples can be adequately described by the mean and variance, that's the condition of normal distribution, and variance of populations are equal or homoscedastic. So that's just a confusing term to say that the variances of the two groups are equal. So if your data does not meet these assumptions, then performing a parametric test may give you flawed results. So there are another class of tests called non-parametric tests that don't make these assumptions. But because they have no underlying assumptions, sometimes it will be harder to find a significant result, which is what we want in statistics. We don't want to come to the wrong conclusion about a significant result, so we're quite cautious. So we want a method that isn't going to incorrectly give us a false positive. So this table is just a bit of a description of some tests you're probably familiar with in the parametric world, such as t-tests, one-factor ANOVA, paired t-tests, factorial ANOVAs, correlations and regressions, and some of the non-parametric equivalents that you'll find in most software. So if you're not sure about meeting the assumptions, often what we suggest is that you try both tests. So if this one gave you a significant result and this one didn't, then you should report this one because you want to be more conservative. You don't want to claim a significant result falsely. So if this one said no significant result, you should report that. If they both gave you a significant result, then you can report the parametric test because it is a more powerful test, and if they're coming to the same conclusion, then that's fine to report this one. And it's very quick to run both tests. I mean, it doesn't take much time to run this analysis in any software. You'll notice there's not necessarily a non-parametric equivalent to every parametric test. So for factorial ANOVA, for example, there's no formal non-parametric equivalent. You can sort of rearrange the data and perform a one-way ANOVA to get around that. There is no non-parametric equivalent of a regression, and a chi-square test. So if we're dealing with count data and we're doing a chi-square test, that is a non-parametric test. And because it's count data, it's not normally distributed, so there's no parametric equivalent for that test. I just want to talk to you about patterns in your data that can lead to misleading conclusions. So this is one of the real benefits of plotting your data before you do anything, really understanding your data before you jump into any analysis, because it's the only way you'd really know then whether the analysis makes sense or not. So if I was to plot this data, the first thing I'd wonder is why I've got this gap here. Have I really sampled two distinct populations, or have I actually sampled this is really one population, but I've actually missed a whole section of the population in my sampling? Now, the reason this could be important is that if they are two distinct groups and I was to fit a correlation or a regression through this data in two separate groups, I would still have a positive linear relationship here in this group, but in this group I would find no linear relationship at all. As values along this axis increase, there's no corresponding relationship along the y-axis here for this group. In this plot here, what we can see is that there is quite a nice strong negative linear relationship in this group of data, but then something happens here and the relationship changes. So I'd need to consider whether there's something going on here. Is this am I measuring a different group of individuals, or are these mistakes? Or should I be not trying to fit just a single linear relationship, but some other kind of non-linear relationship to this data? So in this graph, what we can see is a couple of points on the left-hand side of the x-axis here that look like possible outliers. So something to consider when you see values or observations that you think might be outliers is to consider whether this is just highlighting to you that you are missing sections of your population in your sample, whether these are mistakes or whether they're true events but they're rare events. And that can help you decide whether you want to include them in your analysis or not. If they're mistakes, obviously they should be missed. If they're true but rare events, then what you need to consider is whether your research question is about a population that includes rare events, or you are only interested in the normal kind of response range. What you decide to do with these kind of values is really quite important, because in this example, these outliers actually have really high leverage. So what I mean by that is if we removed these values from the analysis, it would completely change the line we fit to this data. It is these two values that are driving this linear relationship. Without them, we would not be finding this positive relationship here. And if these two values occurred up here in higher on the y-axis, we would actually get a negative relationship. So it's really important when you're thinking about outliers to consider carefully what would be the consequence of removing them and what would be the consequence of retaining them. And think carefully about your research question, about because that can help inform you on whether or not you should be trying to retain them in your analysis. Also, don't be restricted to linear relationships. Often data have quite non-linear patterns, and you need to plot your data first to actually see this. Otherwise, you're going to spend a lot of time trying to fit a single linear relationship that will be non-informative. So if we tried to fit a linear relationship to this data, actually we'd just get a zero slope line. There would be no positive or negative relationship identified from this data. If we try and fit a curvilinear relationship, we get a much better result. So earlier I talked about whether or not you should be using your data for hypothesis generating or hypothesis testing, and the problems with using the same data set for hypothesis generating and testing. This is known as p-hacking, and this is covered in the Model-Litsky paper on the study desk, which you should have a read of. So on the right-hand side here, there's a list of things that you may want to try to do to your data. Things like increasing your sample size, analysing a subset of the data, including more variables in the model, adjusting the data, so maybe standardizing data or transforming the variables in some way, removing outliers, as we just discussed, picking a different group to use as a control, comparing different outcome variables, and using different statistical tests. All of these are perfectly acceptable things to do as part of your analysis. Where they cause a problem is when you do them. So when you analyse your data and you get your result, if it's not significant and that causes you to try all of these different types of adjustments, then that can be a real problem, because the only reason you would be trying these things is to try and get a significant result. If you've got a significant result on your first attempt and you stopped there, you would never know what these kinds of adjustments did to your data and how it may have changed your conclusion. What if doing one of these things led you to a non-significant result? How do you know that is not a truer representation of your data than your first test? So the problem is not having clear questions and not having a clear research path. And so you get your data and just keep digging away and changing and adjusting until you get a significant result. You're chasing after a significant p-value. So all these data modifications are legitimate, except if you only look at them because you haven't found a significant result. You are inserting bias if you follow this process. So increasing sample size, transformation, etc. could turn a significant result to non-significant, but you would never know because you stopped as soon as you got a significant result. If these are legitimate things to try with your data, then why are they only legitimate because you didn't get a significant result to start with? If they're legitimate things to do, then you should consider them in conjunction with your first significant result. So consider these adjustments before inferential analysis. Make sure you do proper exploratory analysis to know whether these kinds of things would be needed. This kind of process is okay if you're just trying to do hypothesis generating. The problem comes when you're doing all of this just for the inferential aspect in the hypothesis testing process. Another form of p-hacking is multiple testing. So it's testing smaller and smaller subgroups of your data until you find some significant result. So this is an XKCD article, and basically they're looking for a relationship between jelly beans and acne, and they test all jelly beans and don't find any relationship. So then they look at each colour independently. So they're doing multiple tests of the same thing, testing the same hypothesis over and over and over again, just with a different subset of data, until they find a result, which is that green jelly beans are the ones that cause acne. Now, the process of doing all of these multiple tests means that each time you do another test on the same, just a smaller subset of the same data, you are more and more likely to find a significant result just by chance. So if you are going to do multiple testing, there are methods to correct our p-values so that we're unlikely to find a significant result just by chance. And what this means is we no longer accept 0.05 as the benchmark, and we will only accept something as indicating a significant result if the p-value is much smaller than that. If you want to look up some of these tests, one of them is called the Bonferroni test, where we take the p-value, 0.05, and we divide it by the number of tests we've performed, and that's our new significance level that we need to reach before we conclude a significant result. So the value of the scientific method is not in p-values, it's reproducible research and repeatability. Scientific or evidence-based decision-making is not based on the results of one study. Our work, our research contributes to a body of work, and when one study after another finds a similar result, that's when we can use that information for decision making. We use inferential analysis to help us make objective rather than subjective conclusions, because the same analysis on the same data will produce the same result if performed by somebody else. Lack of subjectivity does not mean we are removing uncertainty, though. That is why we use probability. 0.05 is a completely arbitrary cutoff point for significance and leads to a false feeling of certainty. So just coming back to one of the earlier slides about whether or not it's important for you to be considering experimental design and data analysis as important aspects of your research. Yes, I think it is, but it's also important to critically analyse other people's design and analysis, particularly if you're using it as a basis or background for your own research. Science is a constantly evolving field. Our methods of data collection and analysis are constantly being improved. So just because something has been published and been used before doesn't mean it should stay static. We can all contribute to improving the way we analyse data and the way we collect our data and the way we present our data. So just to end on another reminder, never to trust summary statistics alone. I've sort of said multiple times it's important to graph your data and visualise your data. So this is an example of just looking at summary statistics. So we've got a mean of an X variable and a standard deviation of that same variable, and then we've got the mean of the Y variable and the standard deviation and a correlation coefficient as well, and our sample size here. So we can get an understanding, well, the means are a bit different to each other on these X and Y variables, and the standard deviation of this one is bigger, and correlation looks a little bit negative, but it's also very weak. Correlation coefficients are between -1 and 1, with 0 being no correlation, so this is just slightly negative. So maybe you're building up a bit of a picture of what this data looks like in your head, but if we actually plot this data, we'll see that we've got a datasaurus. So it's important not to just look at the numbers. You need to visualize your data and look at them together. Okay, so that's all I have for this presentation. Make sure you look at the papers that have been provided to you on the study desk before you start your assignment, and best of luck for the rest of this course and for your research.